Good morning, guys. Today is a very special day because you're going to learn about genomic imprinting and how it came about. To understand epigenetic imprinting, it is crucial to understand epigenetics and its role in developmental biology. Epigenetics is a way to change gene expression without altering the DNA sequence. Instead, you change the accessibility of chromatin. One way to do this is by modifying histones, the proteins that DNA is wrapped around. For example, through histone acetylation, chromatin is decondensed and genes are activated. If you want the opposite of this, you can add a methyl group to the cysteine nucleic acid of the DNA, which will condense the chromatin and inactivate a gene. This hill represents the epigenetic landscape. At the top, we see a pluripotent cell, meaning it can differentiate into any cell types. When an embryo develops, its cells start to lose their pluripotent qualities. They start to differentiate into different lineages and eventually form differentiated cell types. Some such differentiated cells are the human sperm and egg. When they fuse, fertilizations occur, and epigenetic changes are erased so that cells can become pluripotent and develop into all cell types needed to grow the baby. Because of this epigenetic erasal, parents of origin, male or female, should not affect gene expression and function. An experiment to confirm this was carried out on mice. After fertilization, both the maternal and paternal pronuclei, or chromosomes, were removed before they had a chance to fuse. This empty egg was then injected with either two nuclei from the same parent or one of each, as occurs in natural sexual fertilization. Surprisingly, only cells containing a maternal and paternal set of chromosomes survived, meaning that chromosomes differ based on which parent they have been inherited from. This is because of genomic imprinting, where certain genes are expressed differently. Evolution of genomic imprinting can be explained by the tug-of-war model, where the question to how many resources should be allocated to current versus future offspring differs based on sex. Females are related to all of their offspring equally. For them, the children they are carrying now are just one of many clutches they will have, so they will take this into account when allocating their resources. The situation with males is different. Although he is the current father of the children, it is very probable that the female mouse will leave him and find somebody better, making his relatedness to future children zero. This asymmetry between the male and female partners selects on the male to divert more resources to current offspring than is optimal from the female's perspective. This leads to selection for certain genes to be imprinted. This shows how selection will be different depending on whether you're the mother and you want to equalize your physiological investments in each offspring, or if you're the father and you want to coerce the mother into investing more heavily into his offspring. Based on this, the function of maternally imprinted, i.e. paternally expressed genes, will be to obtain as much nourishment as possible for developing embryos above and beyond maternal optimum, for example by increasing placental growth, appetite, and nutrient metabolism. On the other hand, paternally imprinted, i.e. maternally expressed genes, will act to counteract this effect. Further on, we will see some examples.
In mammals, some loci are biochemically marked in order to distinguish between paternal and maternal alleles, and this happens in the testes and ovaries during the production of gametes, which affects the transcription of marked genes in the embryo after fertilization. So let's now focus on an example of how a specific type of imprinting genes works. The hormone we are going to focus on, IGF-2, the growth factor hormone, is widely expressed in the paternal allele, while the mother's allele of IGF-2 is hardly transcribed. This is unusual because according to the principle of natural selection, there should be equal expression of both alleles in order to protect the offspring from recessive mutations. So why does this happen? The answer we can find in the function of IGF-2. IGF-2 is a stimulant for cell division and acts using a cell surface protein type 1 IGF-2 receptor. However, there is another surface protein that also has a binding site for IGF-2 and has a non-growth related function, and this type 2 receptor is only transcribed in the maternal allele. Therefore, the paternal transcribed IGF2 is selected to maximize rates of division in the embryo, while the maternal allele is turned off to conserve resources for future reproduction, and instead the maternal receptor binds to the excess paternal hormone to stop overtranscription and equalize the flow of resources to different embryos. The proof for the effect of IGF2 imprinting can be seen in an experiment done in 1996, where they manipulated the expression of paternal and maternal IGF2. We can see that when there is equal transcription, the embryo is normal, whilst if maternal allele is deleted, the embryo develops as a giant, and vice versa when the paternal allele is deleted. According to Haig's hypothesis, imprinting genes should only be found in groups of organisms that have developed placenta-like structures. Therefore, this form of imprinting only occurs in mammals with placentas and does not occur in animals such as chickens, frogs, and snakes, where the embryos are provisioned before fertilization, and there is no opportunity for the father to manipulate the distribution of maternal resources. Thank you for listening, and we hope you have learned something about how evolutionary natural selection occurs even down to a molecular level in genomic imprinting.